السلام عليكم ورحمة الله وبركاته. درسنا الأول Introduction to Meiosis and Interphase. We have two types of divisions: mitosis for somatic and sexual cells, and meiosis for sexual cells. Both divisions are preceded by interphase. The importance of the interphase is to change chromosomes of one chromatid into chromosomes of two chromatids. The number remains the same, 2n, 2n, but 2n chromosomes of one chromatid becomes 2n chromosomes of two chromatids. Interphase of three phases: G1, which contains chromosomes to n chromosomes of one chromatid; the S phase, where the chromosomes duplicated, each chromosome is duplicated by a process named DNA replication; and the G2, where we have to n chromosomes, each of two chromatids. Also, we notice during interphase the separation la centrosomes, which are important in the division and the formation of the spindle fibers, the mitotic spindle. During the G2 of interphase, the chromosomes replicated during LS. We have two n chromosomes, each of two chromatids, but uncondensed chromosomes. We notice that the cell of G2 has two centrosomes. There is a nuclear envelope or nuclear membrane and plasma membrane. Meiosis. Meiosis is of two successive divisions preceded only by one interphase. Not each meiosis is preceded by interphase. The two divisions preceded only by one interphase. Meiosis 1 of four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. While meiosis 2 is also of four phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 1 is also named as reductional division, while meiosis 2 is named as equational division. During meiosis 1, one diploid cell, which contains two n chromosomes of two chromatids, is divided into two haploid cells. Each contains n chromosomes of two chromatids, while during meiosis 2, these two cells, two haploid cells, are divided into four cells. Each contains n chromosomes of one chromatid. We notice during meiosis 1 in the given figure that the two homologous chromosomes are separated. Each chromosome migrates toward opposite pole to form two cells, but there is an exchange of the DNA genetic material or a segment of the chromosomes between the homologous chromosomes by a mechanism named as crossing over. We are going to study this mechanism later. And during meiosis 2, the chromosome of two chromatids is also separated into two chromosomes. Two chromosomes, each of one chromatid, and each chromosomes will migrate toward opposite pole to form two cells. And we notice the exchange of the genetic material between these two chromosomes. Slide 5 shows the phases of interphase followed by the phases of meiosis. Meiosis is not a cycle like mitosis with interphase. Firstly, during G1, we have two N chromosomes, each of one chromatid. The quantity of DNA equal to C. During S by DNA replication, each chromosome is replicated into, the DNA is replicated to give a chromosome of two chromatids. The quantity of DNA is duplicated to give at G2 a cell contains two N chromosomes, but each chromosome is of two chromatids. The quantity of DNA is 4C. During prophase 1 and metaphase 1, the quantity of the number of chromosomes are 2N. The quantity of DNA 4C, since the cell contains two N chromosomes, each of two chromatids. During prophase 1, we have the crossing over, and during metaphase 1, the homologous chromosomes are arranged at the equatorial plate. Then during anaphase 1, the homologous chromosomes are separated. And during telophase 1 and cytokinesis, the cell membrane is separated by the cleavage furrow to give two cells at prophase 2. Each contains N chromosomes of two chromatids. The quantity of DNA is 2C. Meiosis 2 is similar to that of mitosis. The chromosomes are arranged at the equatorial plate in each cell. Then we notice a separation of sister chromatid at anaphase 2, and during telophase 2, the cells by cytokinesis are separated to produce four haploid cells. Each contains N chromosomes of one chromatid. The quantity of DNA is C. Later, by fertilization, haploid cell from the male and haploid cell from the female, N plus N, give zygote of 2N. Quantity of DNA C plus C becomes 2C. Slide 6 is a simple diagram for the DNA replication during interphase. Two N chromosomes of one chromatid becomes two N chromosomes of two chromatid. Then during meiosis 1, separation of homologous chromosomes to give two cells. Each contains N chromosomes of two chromatids, and by crossing over, the DNA genetic material is exchanged. Then during meiosis 2, we have for daughter cells, non-identical cells, haploid cells, each contains N chromosomes of one chromatid. وظيفتك على الدرس الأول بالميوسيس هو إنك ترسم جراف بيمثل ال quantity of DNA as a function of time اللي هو موجود معك بتابل، واثنين تشرح تطور الايفوليوشن للكوانتيتي اوف دي ان اي، تغير الكوانتيتي اوف دي ان اي خلال الانترفيز اند الميوسيس، يعني بتقول الاسباب اللي عم بتأدي لتغيير الكوانتيتي اوف دي ان اي، بتزيدها وبترجع بتنقصها.